Welcome everyone, and thank you for joining us for today's webinar titled Making Optical and Electrophysiological Measurements in the Brain of Head-Fixed, Freely Moving Rodents. My name is Andy Henton from Inside Scientific, and I'll be your host for today's event. Our session is sponsored by Neurotar. It is going to be a fast-paced, content-rich webinar showing various applications where optical and electrophysiological measurements are being made in conscious, motile rodents enabled by the use of a novel device called the Mobile HomeCage. First, we will hear from Leonard Kirog, CSO of Neurotar, who's going to provide a brief history of the Mobile HomeCage. Following, we will be joined by Drs. Eero Castren and Petri Hyytiä of the University of Helsinki in Finland, who will discuss strategies for optical imaging in the brain and show how they are using this device to study neuronal plasticity. And then we're going to change directions and take a brief look into how researchers can make electrophysiological recordings from the brain in conscious rodents. For this topic, we will be joined by Drs. Roustem Khazipov and Murat Menlebiev of INSERM in France.
Just a brief history of how the Mobile HomeCage came about. We've started a few years ago as an academic lab. In fact, I was a principal investigator for 10 years at the University of Helsinki after being trained as a postdoc at Duke University Medical Center and then later at NIH. And I still have an adjunct professor position at the University of Helsinki as a part-time. But a few years ago, we also started a business service as a contract research, providing two-photon imaging services to global pharmaceutical industry, doing contract research studies, for example, to half of the top 10 pharmaceutical companies and many of the biotechnology companies, and most of the time we of course had to do this in anesthetized rodents. So we had this problem of whether these data that we were generating are really relevant to health and to disease. So there was always this question, and we needed to do research also in non-anesthetized animals. And then by solving this problem for ourselves, we created this device, the Mobile HomeCage, and then we realized that there is also a market for it, so we made it into a product, and now we provide this to laboratories worldwide. We launched this device last year at FENS and then later in the States also at SFN, and this is the story of the Mobile HomeCage. Just a brief overview of what we do is at Neurotar as a contract research organization. Here's one example, trans-blood-brain barrier pharmacokinetics and biodistribution of biologics. In this example, you can see here an antibody that is designed to penetrate the blood-brain barrier and then target the A-beta plugs. Here they are labeled in green with methoxyXO4, and the antibody is labeled with Alexafluor 647. You can see clearly the antibody in the vessels and then how it penetrates and co-localizes with A-beta plugs. In another example here, you can see individual mitochondria in the brain of live mice, transgenic mice expressing CFP in mitochondria, and the blood vessels here shown in red. And then we trombatize individual vessels and create stroke models. And here in this movie you can see non-fragmented elongated mitochondria before stroke, and then after we trombatize the vessels with a laser, you can see fragmented mitochondria really clearly. So all sorts of very fine morphological and functional studies with two-photon microscopy, but of course there's always an issue whether the data are relevant if you do them in anesthetized animals. So this issue was there, and we looked at different solutions of working in awake, non-anesthetized animals. Of course, the simplest solution would be just to constrain the animal or to train it to stay still under the microscope, but we didn't quite like it because it's stressful for the animal to be constrained. We looked at the treadmills. didn't quite like that solution either, because the animal has to move along a single line. There is no no possibility for the animal to choose the trajectory and move in any direction that it would like to move along. There's another alternative for the animal to carry a head-mounted device, like a miniaturized optical microscope, as is shown in this figure here. Here you would have to compromise on the quality of the optics that you can put on the head of the animal because of the size limitation. A very popular solution that is around there in many labs is the airlifted ball or spherical treadmill. We've experimented with this approach. The limitations here are that the animal has to walk on a curved surface of the ball, which is not natural for the mouse, and also that this is an unlimited space that the mouse walks on, so there is no there are no obstacles, there are no walls for the mouse. It continues to move on, and in order to provide the sensory stimulation, the sensory feedback for the mouse, you would need to create a virtual reality around it. And mice are not really visual animals, so they don't rely on the visual feedback as much. This was also not the solution that we were happy with, so we came up with a different solution. We airlifted the home cage. The home cage that the mouse is familiar with, that it feels happy and stressless and cozy in. The home cage that has a flat floor and has the walls. So the principle is similar to the ball, but it's a flat ball with a wall, basically. It has a natural enclosed environment. It allows the extremely short habituation time. It has a very stable head fixation due to the materials that are used here. And it also has a compact design that allowed us to put it under the standard microscope that we use. We had Olympus FluView 1000 in our setup, and this mobile home cage didn't take much space under the microscope. So we were very happy with this solution, and then we made it into a product, and it became available, as I said, on the market about half a year ago. And since then, we've sold about 20 devices around the world, in the States, in Germany, in France, in Russia, in Korea, in Finland, all over the world. These are some pictures, and this is the list of techniques that are compatible with this device. Of course, 2-photon microscopy, whole cell patch clamp, but also optogenetics, intrinsic optical imaging, anything that requires steady fixation of the head while allowing the body of the mouse to move around freely in any direction, choosing any trajectory, navigating around the cage, can benefit from this device. I wanted to share just one example from a reference lab in Munich of Professor Anton Sirota and a senior postdoc in his lab, Nicholas Corrales, who have recently recorded using advanced techniques of combined chronic and acutely inserted electrodes in the mouse. They recorded the sleep states, the sleep and awake states in the mouse. So here you can see the electrodes, the multi-shank high-density silicon probes, and the wire electrodes. These are examples of traces that they have recorded, and some movies. So above you can see a movie of the mouse sleeping in the mobile home cage, and below is the mouse running around. They have a modified head fixation system here, and here are some traces, and then the spectrograms. So here are some examples of the slow wave sleep and the rapid eye movement sleep as well, which of course suggests that the mouse feels quite good if it can fall asleep in the mobile home cage. With this table, I would like to wrap up my part of the presentation, and would like to compare the existing solutions for recording in awake mice. So with head-mounted devices that the mouse has to carry on the head, and then the head fixed on the treadmills, whether these are linear or spherical treadmills, combined with the virtual reality. Both of these approaches require quite long training and habituation times, and are arguably quite stressful for the animals. Both of these are addressed by the Mobile HomeCage. Again, both of these approaches are quite costly. The miniaturized optics are extremely costly. Virtual reality is also quite costly compared to the Mobile HomeCage. The devices mounted on the head of the mouse do allow the mouse to move around in the natural environment on the flat floor with the natural obstacles and objects positioned in this natural environment, whereas the ball doesn't. And this is the reason why researchers would opt for a head-mounted device, for example. On the other hand, the quality of the optics that you can mount on the head are clearly inferior compared to the optics that you can use in combination with the big microscope with a good objective. So these would be then the choice between these two approaches. Again, you wouldn't need to compromise using the Mobile HomeCage between these two. One thing that is missing with the Mobile HomeCage, you cannot work on the place cells because the device is quite small, but we're working on a bigger one at the moment. So we'll be happy to discuss this in more detail if you contact me directly, but with this I would like to pass then the presentation to Professor Eero Castren.
Good afternoon or morning to everybody, depending on where is it that you're located. My name is Eero Castren. I'm at the Neuroscience Center in Helsinki, Finland. And my lab has been working with neural plasticity over many years. There are many different definitions of plasticity. In my group, we're talking about plasticity as a capacity of neurons and neural circuits to change structurally and functionally in response to experience. And what is actually guiding and regulating the plasticity is neural activity. And one of the things that we often forget is that plasticity is not just adding things, but it's also simultaneously removing things. So when you have neurogenesis, you have to at the same time remove neurons through programmed cell death. Axon growth is balanced by selection for retraction of axons. Synaptogenesis is then balanced by pruning of inactive synapses. And synaptic potentiation is balanced by synaptic depression. And through this mechanism, the active neurons and active synapses are selected and stabilized. And this can happen without any net change in the number of synapses or neurons, for that matter, in the brain. So it actually, the plasticity is about quality, not about quantity. So at one sort of part of our study, we were interested in the question of whether anesthesia has an effect on neural plasticity and more specifically on spine plasticity and dynamics. And we had used sections from anesthetized animals that had been then perfused and then cut into sections. And there we were counting the number of spines. And what we see that here anesthesia did not seem to have any effect in this particular brain region, neither on the number of spines nor on the shape of the spines as is shown here, mushrooms, stubby, and philopodia. But now we were also then wondering whether the anesthesia might actually have, if not effects on steady state situation, it might actually have an effect on the dynamics. And for this purpose, you would actually then need to look at image repeatedly. So you would have to take first a microscopy with M here, and then you would go back and look again. Then you would anesthetize animal and then look at the effects of anesthesia either immediately or then long-term after the anesthesia. And obviously, if you wanted to do this, you would have to do where this M is in awake animals. And we were wondering how this could be done, and then we became aware of that just 2 floors up is Leo's lab, and they had just actually found a perfect solution to the problem that we have, and this is this mobile home cage that you just actually were introduced to by Leo. So in this case, you can actually do 2-photon imaging in awake, freely moving mice. And the other important thing is that you can do repeated imaging on identified neurons or neural processes, as shown here as an example from the paper that Leo's lab published some time ago. So we use this system to approach our problem. We were now here imaging somatosensory cortex and identified area in somatosensory cortex repeatedly. And what you can see here on the right-hand side, that we didn't see any significant change in the dynamics of neither formation nor elimination of spines. And here you can see the kind of experimental design that I introduced you previously. So even though in this case our results were negative, I became very convinced and excited about Leo's device, because we are now entering, or we are within the era of connectomics, and here imaging, stimulation and electrophysiology of living brain is becoming more and more abundant and actually necessary. And we are increasingly becoming aware of the fact that anesthesia influences many of those measures that we want to look at. So there's increasing need for recording and imaging in awake animals. And not only only in awake animals, but in many cases in behaving and learning animals. And then if you want to do this kind of experiments, you would like to have the animals in stress, non-stress environment, ideally in their home cage environment. And I feel that what Leo's company has created here actually fulfills many of those criteria. So I think there is a great potential for this system for the future. Thank you very much. From my point of view, I'm now giving the microphone to my colleague Petri Hyytiä.
So good morning, good afternoon, everybody. I'm Petri, and I will tell you a little bit about our small-scale pilot work with morphine. I come from the addiction field, and that's why we actually are interested in the effects of drugs, and in this case particularly the effects on dendritic spines and their plasticity in the brain. What I'm going to do today is just give you a short introduction to the addicted spine, then give you reasons why actually we became interested in the mobile home cage, a couple of words about the hypothesis we are working on, and then describe you the experimental design and some preliminary data of our project, and then I'll try to summarize some of the main points. So when we talk about the addicted spine, we have to keep in mind that what we actually mean with addiction is that addiction is a chronic relapsing disorder, and it's probably marked by long-term changes both in behavior and also in the structure of neurons, especially in the limbic parts of the brain. What I mean by limbic brain regions, I mean the VTA, where the dopaminergic cell bodies reside, and then send projections to the limbic forebrain, nucleus accumbens, and the medial prefrontal cortex. So these are the brain sites that people have been interested in terms of the structural modifications or effects of the drugs. These effects can be very different. For example, in the VTA, people have been recording the size of the cell bodies, but also dendritic arborization, and then spine morphology, and then spine dynamics, especially in the limbic forebrain. So the first demonstration of this kind of plastic exchanges came by Robinson and Culp in 1997, when they actually looked at the effect of repeated amphetamine on the spine density in the nucleus accumbens core and shell regions. And what they found that by repeated amphetamine, they got about 30% increase in the number of spines in both regions of the nucleus accumbens. And when they looked at these spines with multiple heads, they actually got a much larger increase. So this was the first demonstration of this kind of structural changes caused by drugs of abuse, and this has been followed up by many groups afterwards. One of the paradoxical findings is that if you compare, for example, psychomotor stimulants like cocaine, amphetamine, and even nicotine with the effects of morphine, you can see that with psychomotor stimulants, you usually get an increase in the number of spines in accumbens and in medial prefrontal cortex. But with morphine, you usually get in a sharp contrast, a decrease in the number of spines. And this is happening both when you give morphine to the animals, or when the animals are actually self-administering morphine. So this is kind of a paradoxical finding, because, for example, repeated injections of both cocaine and morphine are producing the same kind of behavioral effect. For example, behavioral sensitization, so increased locomotion. So this is really a paradoxical finding. On the other hand, these are all based on cross-sectional work, and that's why actually we don't know much about the dynamics of the spines after repeated drug administration. So that's why people have been interested in a more longitudinal setups where it would be possible to really look at the number of spines, for example, during repeated imaging sessions. And this is just a recent example by Munoz and Cuevas, 1.5-year-old paper, where actually we are looking at the effect of cocaine in anesthetized animals, but using cranial windows. And what they saw here, that during cocaine treatment, there was an increase of spines, both spines, fraction of newly gained spines, and then spine accumulation. And what was interesting that when they looked at the number of newly gained spines and correlated those numbers with the magnitude of cocaine-induced place preference, they got a nice correlation there. So why we became interested in the mobile HomeCage was that we wanted to do this kind of long-term longitudinal imaging and look at the effects of drugs of abuse, in this case morphine. It's very interesting for us because we have no interfering anesthesia. Of course, that's very nice. We don't need any control group because we basically can compare the effects of drugs to the baseline before the drug exposure. What is one of the limiting factors is that we only can look at the superficial layers of the neocortex, in this case the primary somatosensory cortex. That's of course the limitation if you are interested in the limbic parts of the brain. On the other hand, we know that this particular brain region is very well connected with other cortical areas, and thalamus and also striatum. Don't have time to go into the details of all this connectivity that is shown in this figure. And on top, we know that that this particular brain region is activated by drugs, including morphine, shown by metabolic mapping, increased CFOS expression, and also functional MRI. So we started off with the hypothesis that the changes in spine dynamics that we might see are correlated with synaptic activity and serve kind of indicators of the neural or the changes in neural circuitry produced by drugs abuse, in this case morphine, and that the persistence of these changes might point to the mechanisms that are behind the long-term drug effects, for example, craving in humans and relapse in animal models. What would be very nice to do is to correlate these changes with behavior, and actually the mobile HomeCage might also provide some possibilities for that kind of analysis. So just to briefly walk you through this this small-scale pilot here, we have animals that were injected with PBS for 4 times every second day, and in the same manner then after this phase injected with morphine 20 milligrams per kilogram every second day, and then imaged right before the first PBS injection, right after that, and the same way right before morphine, right after the first morphine, and then 2 days after morphine and 2 weeks after morphine. And this is just showing you briefly what you might see in the microscope. This is just imaging the same spot in the some of the sensory cortex repeatedly. First before PBS, then after it, before morphine, after morphine, 2 days after morphine, and 2 weeks after morphine. And this particular movie is actually showing you then, I hope, convincingly that the imaging stability is very good. So on the left side you see the baseline imaging just before PBS, and on the right side you can see imaging 2 weeks after morphine. We are still in the process of analyzing these data, so unfortunately I don't have you the final data, but we are hopefully getting those numbers just in a few days. So just to conclude, for us in vivo two-photon microscopy in these awake animals really yields very interesting unbiased information of drug-induced changes in spine dynamics in this particular part of the brain. We hope that monitoring these changes over longer periods of time will give us more insight into the drug-induced structural plasticity in the brain, and really maybe unravel some of the paradoxical findings that we had before. And I think that this small-scale pilot really demonstrates the feasibility of using this whole mobile HomeCage for this particular purpose, and we are very happy to share this technique with Leo. So I will finish here, and I'll pass on to
Thank you very much, Andy. And we will talk with Marat about the applications of head-restrained recordings in animals using electrophysiological tools. So I will first introduce some techniques that we are using in animals, including extracellular and patch clamp recordings. And Marat later will explain how mobile HomeCage can be used for extracellular and patch clamp recordings. So I will start briefly with overview of techniques that can be used in head-restrict animals. I will start first with an image of EEG recordings from human that everybody of you knows. In animals we don't do much of this type of recordings because we are able in animals to perform something more sophisticated, such as recordings using multi-electrode arrays. So here you can see an example of a 4-shank multi-electrode array in which each shank contains 8 recording sites. So we can insert this array into the brain at desired region of the brain of the rat, which is head restrained. And here you can see an example of recordings with a single recording site from some other sensory cortex of a rat, in which you can, using this technique to monitor local field potentials on each recording site. In addition, you can also detect multiple unit activity. And here you can see an example which is recorded at depths of 400 microns in a response which is evoked by inhalation of fluorosil, which is a epileptogenic agent, so it's an epileptic discharge. And using multiple site recordings, you can look how different layers of cortex behave during this epileptic activity. After that, what can be done is that all these local field potentials can be averaged, and we can identify sinks and sources of the local field potentials which are generated during epileptic activities but also during physiological activities. And from the multiple unit analysis, we can identify which layers are driving these epileptic discharges. For instance, in this case, we can see that population spikes are driven by firing of infragranular layers, and with later recruitment of supragranular layers, and with very much delayed and much weaker activations of layer 4 cells. So then in more sophisticated configuration extracellular recordings, in this case the throat is presented, so these are 4 recording sites which are placed into the brain, and each of these electrodes is detecting action potentials from the neurons which are located nearby this recording tetrode. By comparing size and amplitudes of the spikes on each recording site, we can extract from multiple unit activity spikes of individual cells, and through this clustering analysis we can identify neurons. So for instance, here is shown a class of a cluster of spikes which belongs to a single neuron, the way how you see it from different 4 electrodes. And the advantage of this technique is that it enables you to record simultaneous activity of several cells and relate this to network-driven activity. So that is all about extracellular recordings. I will switch now to patch clamp recordings. Patch clamp recordings have something else than extracellular recordings. They enable to monitor synaptic currents which are generated in individual neuron, and so synaptic currents that control activity of cells. In voltage clamp mode with low chloride, that's the usual technique that we are using, we can separate GABAergic and glutamatergic currents. So here you can see an example of traces recorded from a single cell at different holding potentials that enable you to see GABAergic and glutamatergic component of sensory evoked response in layer 4 cell of a rat which is 3 days of age. So you can see here that with a short delay there is a gamma oscillation which is built by individual AMP receptor mediated currents, and then you can see also a summation of this currents which is largely mediated by NMD receptors. At the same time, when you hold cell at reversal of glutamate, you can see that GABAergic currents are generated with a much most important delay after stimulus, and basically GABAergic currents curtail the discharge. So this technique thus enables to go into more details to determine synaptic mechanisms which govern functions of individual neurons. And in current clamp mode, we can record firing of single neurons, and also to study subthreshold conductances, which are equivalent to what is shown on the right side in voltage clamp. So then, in addition, another application of patch clamp can be recordings of neurons in cell attach mode. So in this case, we establish gigasil contact with the cell membrane, and we can record action currents from this cell, but also we can also record activity of single ionic channels in this membrane. So here you can see an example of GABA A channel openings activated by GABA, which is put into the pipette solution, and so all these downward deflections are corresponding to opening of individual GABA channels. And here you can see a simultaneous activation of 2 GABA A channels. The advantage of this technique is that it enables to determine the reversal potential of GABA response and driving force for GABA to study the polarizing or hyperpolarizing actions of GABA in cells without affecting intracellular chloride, which is interesting for various applications, including development, epilepsy trauma, etc. So after this overview, I'm giving word to Marat, and he will talk about the use of these techniques that I have explained in animals in Mobile HomeCage. Marat, please.
Hello, can you hear me?
Yes, we can, Marat. Yep.
Oh, cool. Okay, so this is an example of one of the recordings. The experiments were performed in adult mice P32. So we did several experiments. So this is from P40 mice. Mice were trained using Mobile HomeCage during 1 week, and we applied head holder that called as a helicopter because of its shape on the area above the hippocampus. And so then we used 6M channel silicone probes that was nicely explained by Roustem. So the details that neighbor electrode plates, they are separated by the distance about 1/10 of the millimeter, so about 100 micrometers. And the example of 10-second duration trace is shown here. You see black traces that correspond to the deflection of local field potential, and we inserted silicone probe in the C1 region of hippocampus. So at this depth is a Stratum Radiatum, and a little bit above you will find the Stratum Pyramidale. Black traces, as I said, it's a deflection of local field potential, and the red bars are multi-unit activity that we can extract from our recording. And on the background I put the result of current source density analysis. Due to this analysis we can find, or we can say that at some points, something like here, you see cold colored spots. We have depolarization of the cells, whereas hot colors correspond to the hyperpolarization of the cells. And yet to characterize deflections of local field potential in frequency, we did a continuous time frequency analysis, continuous Fourier transform for the Stratum Radiatum, and we found that there is plenty of theta activity here, but sometimes the theta is associated with a burst of gamma oscillatory episodes. So you see this spot that corresponds to the very fast oscillation in gamma frequency range. Level of coherence between this gamma and theta episodes was calculated using cross-frequency coupling. So theta is on x-axis, whereas y corresponds to gamma. No, it's another frequency range, but you can find that there is a positive correlation between theta frequency band and gamma frequency band, more than 40 up to 100 Hertz. And pool data demonstrates that there is a very nice linear fit between power in theta band and gamma oscillations. So more stronger theta we have, much more stronger gamma will be associated with this episode of theta oscillation. We have also calculated the phase preference for multi-unit activity. So we used multi-unit activity in Stratum Pyramidale and Stratum Radiatum, and we looked for the phase preference to deflections of local field potential in Stratum Radiatum. And you can see that there is a very nice phase preference between units in Stratum Radiatum and its local field potential, the local field potential in this layer. So you see peak at -1, so it's something like -60 degrees from the negative peak from the negative trough of the theta cycle, of this gamma cycle. And the same, we have found nice high value of phase log between multi-unit activity in stratum radiatum and theta cycles. Whereas in stratum pyramidalis, despite of the fact that we have seen much more units, this unit demonstrated very low level of phase preference with deflections of local field potential in stratum radiatum. And finally, what we can do with mobile HomeCage also, we can record from single cell. A few second duration episode is shown here, and here it's rescaled. You can find 2 spikes, traction potential. So the cell was clamped, and this cell was recorded by Pepin Marshall in Helsinki. So the cell was recorded in current clamp mode, and it was kept at minus 76 millivolts, and you see those 2 action potentials. So it's possibly. And as far as I remember, the total duration of this recording was 53 minutes. So it's extremely long. It means that this preparation is very stable. That is amazing and very promising. But for the users, for future users of mobile HomeCage, I would like to advise, to provide some advices. So it's better to find the right coordinates of the structure you want to patch or you want to record, because during the experiment it's impossible, because you have to fix the head holder before training and before the experiment. So it's better to leave the area you want to patch through inside this inner window, because if you would like to record activity or to patch cells outside this inner window, you'll see the problem of pulsations. Another point is that it's better to cover the cortical surface with agarose to keep it wet. Otherwise, you will get some kind of membrane on the surface that will break the tips of the pipettes very easily. And of course, to escape the problem of pulsation, to ensure mechanical stability, it's better to make the hole as small as you can, as small as possible. And it helps a lot to keep the stability, putting the cement around this hole. So you will fix the bones, thus you will decrease the pulsations amplitude. Animals, you can also minimize pulsation using agar or silicone oil if needed. So you just put a drop of agar or silicone oil inside this inner window. It's really difficult to patch a lot of cell through one hole because you damage the tissue, and the maximum number of penetration you can do, it's something like around 10 penetrations. In our case, we used vertical penetration, and it was okay. So we did plenty of gigaseals, and I think that vertical penetration is the best solution because of this cone shape of the pipette. When you insert the pipette, you press the tissue, the cortical tissue, and so you minimize. You also minimize pulsation. And finally, depending on your structure, it's better to have the pipette with exact length of the tip, because if you will have very long tip of the pipette, you will have extra capacitance that is not good. And for example, in mice, in adult mice, for patching in stratum, the tip length is about 3 millimeters. Okay, that's all, and I think that we have to move to the Q&A section now.
And all right, so first question. We've had a couple questions come in that talk about habituation time and also the training protocol for the animal. Marat, also you used the term the helicopter stage. That would be the part that attaches to the skull. Could the group elaborate on that? Maybe Leo, if you could take the lead on discussing, the technical considerations for setting up an animal for use in the cage, and then our users could comment on their experience?
Yes, sure, I'll be happy to. So the helicopter term refers to the metal bar that has the shape of a helicopter or a butterfly. This is usually attached with dental cement to the animal's head in advance, about a month prior to recording or imaging. Then the habituation of the animal and training in the mobile home cage usually takes 3 to 4 days prior to imaging or electrophysiological recording. And this is extremely short compared to other head fixation devices such as the spherical treadmill or the airlifted ball that usually takes 10 days or more according to our experience and the experience of other users using the ball. So this is what we refer to when we say that the habituation time is short.
As an extension of that question, how long can you actually make recordings or take images once working with the animals in the home cage environment?
Roustem, would you like to take that question?
Well, we usually go for a day, yeah? Actually, we have limited experience with mobile home cage, but for the rats that we are recording with the conventional head restraint situation, what we do, and we also work with baby rats, so it's quite different situation. But in our case, what we usually do, first of all, we prepare animals for recordings day before we start recordings. Then the wound heals, and on the next day we fix animal head. Then during recordings we feed animal regularly each 15 to 30 minutes with artificial milk, and animals feel quite happy. And usually our recordings last for a day, let's say. We have also made attempts to do it during a couple of days, and it works quite nice, I must say. But we have never tried for doing it for more than 2 days. Now with mobile home cage, I think that it can go even for a longer time, but we'll have to think about a device which will provide feeding and water to the rat or to the mice, which is in the mobile home cage. Something to work on.
Yes, but our experience with the mobile home cage so far has been up to 2 hours. We provide the water to the mouse, but if water can be provided in the automated way, then it can stay there for longer periods of time. It doesn't feel distressed. And now that we have evidence that it can fall asleep in the mobile home cage, then it's clear that it can stay there for longer periods of time. So chronic imaging, for example, in the epilepsy research or wound healing or stroke research is clearly conceivable and feasible.
Excellent. And actually, I think on that note, a question has come in about just any tips and tricks for preparing and managing the cranial window in those chronic applications.
Mm-hmm.
So can you offer any advice on how to, kind of maintain feasibility of the imaging site?
The standard protocol for implanting the window is that we prepare it 1 month in advance, mm-hmm. and we carefully remove the bone and without disturbing the dura mater, and then we replace it with a glass and fix the metal bar there with the dental cement. So it's a pretty standard protocol, okay. Which then results in half of the windows approximately recovering the transparency and half remaining opaque in mice. It never works in rats. Nowhere in the world it does. All windows in rats then go opaque and stay opaque, unfortunately. Okay. So chronic imaging is not possible in rats, unfortunately.
Okay. Very good answer. And just speaking about rats, we've had a question come in about suitable. Most of the applications discussed today were discussing mice, but other smaller rodents. Are there any people using rats, maybe gerbils or other small rodents? Is there experience there, or can you confirm that users can do this?
On Mobile HomeCage, in the standard size that is available now, it only works for mice. For rats, the MHCL, Mobile HomeCage Large, is going to be released in a couple of months, hopefully. It's being designed in the 3D. It's already available, but not in the metal and plastic yet. So it's going to be almost twice as large, and then it will accommodate rats and small rats up to 300 grams as well.
Great. Okay. Very good. Also, talking about imaging, what about the max imaging depth? I'm not sure if this is actually dictated or relying on the home cage or just more the application itself, but we've had this question come in from a couple people.
It's the same as in anesthetized animals. In the cortical window implanted flat on the surface of the cortex, you can go as deep as your 2-photon microscope will allow you, which is typically up to 1 millimeter or 1.1 millimeter. If you're using hippocampal windows with part of the cortex removed, then you can image maybe 500 microns into the hippocampus. But these are the typical depths that you can do with 2-photon microscope.
Okay, great. All right, we've had some other questions come in that I think are focused on behavior paradigms or studies. So I'd like to ask you first, in your experience, is anyone looking at using the cage for different behavior paradigms where users would also be able to look at frequency of activities and rewards and nose pokes and things like that that are common to other home cage is the term used systems for operant conditioning and behavior test.
Yes, there's several groups using Mobile HomeCage for combining behavior with imaging or with electrophysiology, and some of the proof of principle experiments are shown in this Jove paper that Eero has referred to in his talk. So if you review the recording of this webinar, you have the reference to that paper. There we have the novel smell recognition paradigm. Where the smell-presenting cotton swabs are attached to the cage walls, and we then record the amount of time that the animal spends next to that cotton swab. Other paradigms could be with auditory stimulation or light or again smell, putting novel objects into the cage. Other behavioral paradigms that people are doing is, for example, putting a second mouse, non-head restrained mouse, into the cage and looking at social interaction, courtsmanship, dating, a mouse of an opposite sex in the cage, and then vocalization related to social interaction. So all sorts of things. Mice are happily behaving in the cage, and they can be stimulated, and they can also reveal the behavioral feedback or develop behavioral phenotypes in the cage, because they can move freely while the head is firmly fixed for recording or imaging.
Excellent. And actually, that comes to a question that's fitting, and also, Leo, you mentioned this during your presentation, somewhat commenting on the confidence of that, Neurotar feels very confident that this does not induce a large amount of stress in the habituation and in the measurement field, that the home cage itself is a very accommodating and therefore an optimal choice. Can you just kind of reiterate or comment on that again, share your thoughts, because we've had a couple questions come in from the audience about, stress and training, things like that.
Yes, so what we've looked at, so I've shared the data on the sleep, which is very reassuring. Then we've looked at the weight loss during the training. There's an initial weight loss of about 6% on the first day of training, which then quickly recovers on the third and fourth day. It's a full recovery of weight of the animal. The amount of movement, all these data are in the Jove paper. The general behavioral expressions that are typical of the stress, such as vocalization or the tail erection or whisker movements, etc. So judging from all of that and the feedback that we get from the users indicate that there is a very quick accommodation to the home cage on actually the very first day of training. As soon as you put the animal in the cage there, it very quickly gets used to moving in the cage without any use of anesthesia on the first training day, which is again different from other devices.
Right, right. Excellent. Well, in the interest of time, I'm going to say that we'll close our Q&A, at least the live session for our Q&A now. Reminder to everybody in our audience, you will be greeted with the option to fill out a survey after this event. So please take a couple minutes to complete that and give us feedback, you know, on the content of today's webinar and maybe what you'd like to see moving forward as it relates to photon imaging and electrophysiological measurements in the brain of awake and motile rodents. I will thank our guest speakers today. To all of you, thank you very much for joining us, our group from Finland and our group from France at INSERM. And a big thank you to our audience that have logged on for the live session. We hope to have you back again at a future session soon. Feel free to continue sending in questions. Again, everything will be wrapped up and included in a transcribed copy in the Q&A report. So look out for that in the coming days. You will receive access by email to these resources at that time. So again, thank you, our guest speakers today, and have a wonderful day, everybody.
I'd like to join you, Andy, also for in thanking the speakers for taking their time, and thanks to the audience.
Yeah, thank you.
Thank you.
Yeah.
Wonderful. Have a great day, everybody. We'll see you again soon.